Alright, so this is going to be just like the activity you're going to do next. So I made the notes almost exactly like the activity, so this is going to be also an example, so that's why I put modeling meiosis example at the top of the notes. So at the top I have this key. Now I use different genes than what you're going to get in the example. Alright, so at the key you can just look at these. You actually you'll have to fill these out. So again, just remember the big A is dominant, little a is recessive. These are the phenotypes, and then our genotypes are the letters. So whenever you have a capital letter, you get the dominant phenotype. Whenever you have two little letters, you get the recessive phenotype. So these are all the possible genotypes and phenotypes that we have. Alright, so here's our original cell. We have heterozygous, big little, for A, which is attached earlobes. We have, and what I'm going to do is I'm going to start actually writing these down here because it says what are the genotypes. So we have big little, and that's attached. We have big little R, and that is widow's peak. Which is like a the hairline, you have a little peak. D, big D, little d, again heterozygous, and we have no dimples. And then finally we have little h, which is no hitchhiker thumb. Alright, so there's our genotypes and phenotypes. So the first thing we're going to do is we're going to start with the original cell. So normally our cells have 46 chromosomes, but we're going to start with just 4. Alright, so these are the 4 chromosomes. So when you cut these out, you'll have the chromosomes, and when you come out, don't come out perfectly. You see how I have a little bit of edges? Alright, and you just need 4 of them. I have 8. I have 2 of each chromosome, and there's a reason for that. But right now you just need 4. And so this is your cell. So you can have this set up on the side of your paper. Because my viewing space is only so big, I have it right on top. Alright, so these are my 4 chromosomes. I want to make it just like this original cell. So that's what it is. I have one white one and one gray one. One white one and one gray one. You notice how they have the same genes on it, but maybe different alleles. These have the same alleles because they were homozygous recessive, but these all have different because they were heterozygous. So what I'm going to do is I'm just going to quickly draw that in my original cell. Alright, so how many chromosomes were there at the beginning? There were a total of 4. 1, 2, 3, 4. Alright, so this is the cell out here, and these are the chromosomes. Alright, so the first step in meiosis that you guys looked at in the process is to replicate the chromosomes. That means to copy them. Alright, so I'm going to show you what the chromosomes look like first. So here's my original cell, and then what we're going to do is we're just going to make a copy of each. Alright, so this one duplicates and makes the exact same copy. That one does. This one and this one does. Alright, so now we have a total of 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. These 2 are the same, these 2 are the same, these 2 are the same, these 2 are the same. All they did is DNA duplicated, so you got the exact same genes on those chromosomes. So I'm just going to draw that real quick. So now, how many total chromosomes do we have? Well, 4 times 2 is 8. 1, 2, 3, 4, 5, 6, 7, 8. Alright, I'm just going to quickly draw our alleles on here, our genes. Alright, so now we can turn the page. So, in meiosis, the cell actually divides twice. So the first thing we want to do is divide once. So first I'm going to set up all my chromosomes here for you. Put the copies in here. Alright, so there's all my chromosomes. So the first thing they do is they divide once. So it really doesn't matter exactly how you divide them. So what I'm going to do is just say these guys are going to go in one cell, these guys are going to go in the other cell, these guys are going to go in one cell, these guys are going to go to the other. Well, actually, I'm going to do it this way because this will matter a little bit. All right, so instead of just, I'm going to take basically one from each pair in one cell and one from each copy in the other cell. All right, so we would have 4 in each cells, and it's basically right now just like our original cell. So this is almost just like mitosis. All right, so remember mitosis made 2 cells that were identical to the original cell. Well, that's pretty much what we did right here. And move those cells, those chromosomes, so now we have 2 cells. So how many chromosomes are there in each cell? Now there's 4 in each cell because we split them. So now meiosis takes it one step further and we divide them again. All right, so now this is where I'm going to show you where we can have a bunch of different possibilities. So I might take the 2 white ones on this one and put it in that cell. And I'm going to take these 2 gray ones and put in this cell. But on the other side, let's say this gray one goes with this white one, and this white one goes with the gray one. So, now first of all, how many chromosomes do we have in each cell? We have 2. All right, 2 chromosomes in each cell, and that's because this is important because later on these are going to be used for fertilization. All right, and now also if you look at, they're all different. This one has a big A and a little big R. This one has a big D and a little H. The other one that has this chromosome in it has a little D and a little H. So these 2 are different, and these 2 are different, and they're all different from each other because they all have different letters. So I'm just going to draw these chromosomes in here. So this one had a little A and a little H. This one had big D and big D, little D and little H. Oh, sorry, little R. So this one actually got all the recessive alleles. This one got little d, little h, and then a big A, big R. So it got the 2 dominant alleles on that one chromosome and 2 recessive alleles on that one. This one got a big D and a little h, and this one got a little R, little A and little R, and this one got the big A and the big R, and this one got the big D and little h. All right, so what are these 4 cells called? Well, these cells are called gametes, and all the cells are different. All right, so that's the difference between mitosis and meiosis. Is meiosis makes 4 cells that have half the number of chromosomes. Remember, we started with a cell that had 4 chromosomes, and then we split it into half. So it creates a cell with half the number of chromosomes. These are called gametes, which are the sperm and the egg. All right, so if this was a male, these would be sperm, they would have little tails. If it was a female, these would be eggs. All right, and they're called gametes, and then these cells here are going to be used for a process called fertilization, and that's when the sperm and the egg meet. All right, in human cells, there would be 23. So remember, our original cells have 46 chromosomes. Well, these gametes would all have 23 chromosomes, half as many. All right, we only use 4 chromosomes, so we made 2. This one would have 23. This is so during fertilization or sexual reproduction, the offspring will have the correct number of chromosomes. All right, because if we had, if we went through mitosis and we were stopped here at 4. When these 2 combine and they each had 4 in them, then the baby would have 8 chromosomes, which would be twice as many as we would want. Put it in human terms, this one would have 46 here. So if this was a human cell, that would have 46, and then we combine 2 with 46, and you would have 92 chromosomes, and that baby would not be functional. All right, so that's part 1 of that modeling meiosis.